Hello students, welcome to Bio Whisper, my bio podcast channel. Today we are going to start the second lecture of Animal Kingdom, which is one of the most important chapters of class 11 biology. So Animal Kingdom have been characterized on the basis of some traits. So what is the basis of classification? At first it's levels of organization, then it is the term of symmetry. After that we have germ layers, then coelom. After coelom, it's the digestive system, and finally we have the segmentation. So levels of organization, symmetry, germ layers, coelom, digestive system, and segmentation. Let's study them one by one. So levels of organization. There, we all know there are four levels of organization: cellular, tissue, organ, and organ system level. So cellular level, the example is porifera, tissue level, nitidia, ctenophora, organ level, platyhelminthes, and after platyhelminthes, it is the turn of ascahelminthes and others that follow the organ system level. So cellular level, porifera, tissue level, nitidia, ctenophora, organ level, platyhelminthes, and organ system level, ascahelminthes and others. Now symmetry. What is symmetry? When the body is divisible into equal halves, that is known as symmetry. When the body is not divisible into any planes into equal halves, it's called asymmetrical. When the body is divisible into any equal number of planes while it passes through the center, it's known as radiosymmetrical and bilateral symmetrical. When we can divide something here only right and left, that laterally, it's known as bilateral symmetry. So sponges asymmetrical, radial symmetrical. We have echinodermata, that's the adult part, nitidia and ctenophora. Bilateral symmetrical, echinodermata and dermata, that's the larval stage only, and platyhelminthes and others. So asymmetrical sponges, radial symmetrical, nitidia, ctenophora, echinodermata, only the adults. Echinodermata, larva, these are bilateral symmetrical, and platyhelminthes and others. Now we have the germ layers. What are germ layers? These are the primary layers of cells formed during embryonic development. During the development of embryo, primary layers of cells are formed. Later on, these develop into tissues and organs. So we have the outer layer, ectoderm. It forms skins. It forms skin, epidermis, outer layer, nervous system, hair and nails. Then it's the turn of mesoderm. The middle layer, it forms muscles, bones, blood, heart and kidneys. And the innermost layer, it forms the digestive tract lining, livers and lungs. So what are germ layers? These are primary layers of cells. Germ layers are primary layers of cells that are formed during embryonic development. Later on, these develop into tissues and organs of the body. The outer layer is known as ectoderm. It forms skin, nervous system, hair and nails. Mesoderm, that's the middle layer. It forms muscles, bones, blood, heart, kidneys. Endoderm, it forms the inner layer, and it also forms the digestive tract lining, liver and lungs. Based on germ layers, animals are either diploblastic or triploblastic. Diploblastic, that means they have two layers, two germ layers, ectoderm and endoderm, and in between there is an undifferentiated cell structure that is known as mesoglea. Now, triploblastic, we have ectoderm, mesoderm as well as the endoderm. So the example of diploblastic organisms, Nitidia, Ctenophora, and after that platyhelminthes and other onwards, They follow the triploblastic germ layers. So Nitidia and Ctenophora, diploblastic, Platyhelminthes and other onwards, they are triploblastic. Podifera doesn't have any true germ layer. So there is no question of endoderm, ectoderm or mesoderm over here. Now the third one, what is coelom? What is coelom? Coelom is the fluid-filled body cavity which is lined by mesoderm. So based on coelom, the presence of coelom, these are of three types. Acoelomate, we don't have any body cavity. Pseudocoelomate is false coelom, and eucoelomate, that means true coelom. The examples of acoelomate, platyhelminthes, pseudocoelomate, ascalominthes, and annelida and others, that's eucoelomate, that's having true coelom. Now let's see the structure here. Here we have the gut. Here we have the gut. After the gut, it's the endoderm. After endoderm, it's the mesoderm, and here's the ectoderm. There is no cavity here. There is no cavity. There is no cavity here. So gut, where endoderm, mesoderm, and that one the ectoderm. We have no cavity here. So it's called, what is it? What is it called? It's called acoelomate. An example, sponges, uh, platyhelminthes, sorry, example acoelomate, platyhelminthes, pseudocoelomate. So pseudocoelomate. Now here we have the gut. Surrounding the gut we have the endoderm, and endoderm is mesoderm. And here there are certain pouches like a structure which are known as mesodermal pouches. And this is the outer layer, ectoderm. Now let us look at the picture of this one. That's the most true coelom. What is true coelom? Here we have the gut. Outside the gut it's endoderm. Then we have the ectoderm. From here it's the outer layer, ectoderm. Here ectoderm, mesoderm. So here we have mesoderm, and here too we have the mesoderm, and in between we have the red colored cavity. So that body red color cavity is, which we have depicted in red color, the cavity is known as coelom, and it is lined by both sides. Here we have the mesoderm, which is by mesoderm. So platyhelminthes, acoelomate, ascalominthes, pseudocoelomate, here pseudocoelomate, and that here the tricoelomate. Platyhelminthes, acoelomate, ascalominthes, pseudocoelomate, and eucoelomate, and annelida onwards. Now let us study about digestive system. So, or alimentary canal. So we have here the elementary digestive system, incomplete digestive system and complete digestive system. Incomplete digestive system has only one opening for ingestion, for taking in food, and also for excreting waste products. Example, Nidiria, Ctenophora, and also Platyhelminthes. Nidiria, Ctenophora, and Platyhelminthes. And here we have the complete digestive system. That means two openings, one for taking food, that's mouth, and one for egestion, taking out waste products from the body. And the example, Ascalminthes and onwards. So Ascalminthes has the complete digestive system. Segmentation, body divided repeatedly. Here the body has been divided repeatedly. Segmentation, body divided repeatedly into repeated segments or parts, externally as well as internally. This phenomenon is known as metamerism. Annelida, we have here two segmentation. Body is divided into repeated parts externally as well as internally. But in Arthropoda, only external segmentation is found. So, when body is divided repeatedly into repeated segments or parts, externally as well as internally, the phenomenon is known as metamerism. In Annelida, we find true segmentation, while in Arthropoda, we see only external segmentation. Now, the first time appearance. The digestive system at first found in complete digestive system first found in Ascalmanthus, circulatory system Annelida, and reproductive system first found in Arthropoda. So that's it for today. We will continue with this lecture series another day. Thank you.